Good afternoon, everyone. Thank you for being present in person, and people who are attending this virtually, thank you for all the comments that you're making on the chats. If you have any questions, you can just put it on the chat box and I'll try to answer them there. So the talk today is the role of mouse liver lncRNAs in Nash and liver fibrosis that are being identified by single-cell transcriptomics-based gene co-expression or co-expression regulatory network analysis. So let me give you a brief background of the disease that we are looking at here, that is Nash. So Nash basically starts with a simple fat accumulation, which is known as steatosis, that eventually progresses into a more serious condition, which leads to excessive inflammation and cellular damage, which is called Nash. And then there are more severe conditions that leads to extracellular matrix deposition, which is called liver fibrosis. And it is one of the second most common reason currently for liver transplantation. And the molecular mechanisms are currently not well understood for Nash and fibrosis. And currently there are no effective treatments, hence the reason for interest. A little bit about lncRNAs, which is also the part of the title. For people who are listening to this for the first time, non-coding RNAs or lncRNAs are usually RNAs that are 200 base pair in length or more. They are non-coding, as the name suggests. And they transcribe from either RNA polymerase 2. That means that they can undergo alternative splicing, and they can form secondary and tertiary structures. But what they are different from a regular mRNA is that their abundance or their relative expression is relatively low as compared to the regular mRNA. And they're highly tissue and cell specific. And that can help you to understand why I'm using the single cell aspect in this regard. Why are they important? Well, there have been studies that have shown surprising amount of diversity for long non-coding RNAs, even though they are not coding. They have upstream regulation that they can form in form of decoys, scaffolds, or guide and enhancers. That means they can bring multiple DNA binding proteins, RNAs, etc., together in the same platform, and their tertiary structures can actually guide that interaction. And hence, they can be essential, very important regulators, as we have seen in case of exist, need, one, and many more. Let's talk a little bit about liver. So liver is highly complex organ that performs a lot of functions. Hence, it exhibits basically this phenomena of a metabolic zonation, which means that within the same cell type, you can have different gradient of expression. That means you have one pericentral region, which is this extreme over here, which is responsible for functions like xenobitic metabolism, gluconeogenesis, etc. While there's this extreme end, which is highly oxygen-rich region of periportal hepatocytes or periportal region, which is responsible for gluconeogenesis or biosynthesis, etc. Hence, there are these complex functions that are changing in terms of gene expression that are different across within the same cell type. You can observe that. If I talk about recent advances in single cell technology, we have been able to explore a lot within the past few years. There have been incoming of a lot of data sets that has helped us to understand an homeostatic liver, that healthy liver comprises of hepatocytes and non-hepatocytes consisting of endothelial cells, the stellate cells, and other immune cells. If I talk about Nash, we have observed differences in zonation, and I'll be showing you some results regarding that we see a difference in shift in zonation in endothelial cells. And also there is this emergence of a very specialized Nash associated macrophages that emerge from the macrophage expansion. And then there is also recruitment of monocyte-derived macrophages. If I talk about liver fibrosis, the model that we're looking at here is a chemical-induced CCL4 exposure that leads to fibrosis that results in the hepatic stellate cell zonation differences. And then there is also the very central hepatic stellate cells that lead to these collagen-forming myofibroblasts, and those are the cells that are responsible for developing those extracellular matrix of collagen and responsible for that fibrotic niche, as we say. But there's still a lot of missing piece of information here with respect to lncRNAs, and that's what I'm investigating here. So basically, what are the lncRNAs that are detected in these different cell types? And which of them are actually responsible in Nash and fibrosis? That helps me to formulate my key biological questions here, that which lncRNAs are dysregulated in Nash and fibrosis, And in this case, I'm using a diet-induced model and a TCT, which is basically a chemical exposure model, to actually look into lncRNAs in more detail. And also, what differences do we observe in terms of spatial zonation between a healthy and a diseased liver type. And eventually, what we do here is lncRNAs, the one that we have, do not have functions associated with them. So we use regulatory networks to actually understand which of these lncRNAs can have essential functions in regulation, both in control and disease. Just a quick review about the analysis strategy here. So for the discovery of lncRNAs, since they're highly specific, we have our own comprehensive list that we have discovered. And the way to do that is basically use bulk RNA-seq datasets. And I have used about 2,000 bulk RNA-seq datasets across 68 different conditions that has helped us to find that set. And then all the analysis for single cell has been done using public datasets that have been published in past few years that we have accumulated. And all the downstream analysis for pre-processing of the datasets we have done in Cell Ranger. Harmony has been used for cross-batch correction. And the spatial or trajectory inference analysis, we use packages like Model and Scorpius and Parallel. And for network analysis and target identification, I'll be going a little bit more into detail later. We use Big Scale 2 for network construction and a sequence context method called triplex domain finder, TDF. Helps us to identify putative targets for some of these regulatory lncRNAs. So the first section, which is basically to discover novel lncRNAs, we, as I said, used about 2,000 bulk RNA-seq datasets to assemble the transcriptome using two methods, Tucker and Huffman, and we have this comprehensive set of 48,000 mouse liver-specific lncRNAs. Some of them have well-characterized functions, but most of them that we have here are quite novel and we do not know what they do. And we use this particular set to actually use as a guide and then use as a reference for all the single cell analysis annotations that we have done in the study. Let's look at these lncRNAs from a diet-induced Nash mouse liver model. So the model that I'm using here is basically 34 weeks of amylin diet that leads to a Nash high condition. And what you're seeing here is basically the UMAP consisting of 11 different cell types from that particular data set. And some of the major findings here is that we find about a dozen of these lncRNAs, about 862 lncRNAs that are highly perturbed across different cell types. And here I'm showing an example of endothelial cells. And another important feature, as I mentioned, was Nash, which is Nash associated macrophages. So what happens here is that your Cooper cell population can further split into these subpopulations. And of this, the one that you see in the middle is called NAMs. And they are the ones that are used these days for investigations are being conducted to use this as a therapeutic target because they're associated with that particular disease phenotype. And we find about 100 lncRNAs that are essential markers for Nash associated macrophages, which can actually also be, we can think about them as biomarkers that can be developed in the future. And then the third finding that we have here is a shift in zonation. So as I mentioned, that endothelial cell, we know that endothelial cells basically show zonation from phase central to phase portal region, as you can see in case of the homocysteine chow diet. And what we see in NASH is that there's a shift in zonation for a lot of these lncRNAs and also protein genes where they change their behavior in their expression and then suddenly are in the phase portal or phase central or vice versa. And these opposite responses are being shown here as an example. In section 3, we are talking about a chemical-induced liver fibrosis, where I'm using basically a healthy liver and a CCL4 6-week exposure resulting in a fibrotic niche. And that here, this particular data set is specifically designed for mesenchyme. That means you're looking at only hepatic stellate cells and the two population cells, muscle cells and fibroblasts, that forms the mesenchyme. The reason being that that's actually, even though it's a minor subpopulation, it's the one that's responsible actually for HSC activation is responsible for fibrosis. The key findings here again is that we have about 200 lncRNAs that are perturbed across hepatic stellate cells. And we see again a shift in zonation that we see from healthy to fibrotic niche. And we see the patterns have been changed. And from that aspect, we also extract these pericentral fibrotic cells that actually then further from the resting stage are differentiating into this collagen-producing myofibroblasts. And we find a lot of these incarnates are actually responsible markers for myofibroblast or collagen-producing cells. That means they are only expressed in these when you have that pathogenic condition. Now, looking at the third model, which is the TCDD, it's a little different model. Here we are looking at TCDD, which is an environmental chemical, but it's also an AHR agonist, and it promotes Nash and fibrosis in mice. That's what we know. And we are looking at lncRNAs in respect to TCDD, and this particular study is a single nuclei RNA-seq as opposed to single cell. The reason being that our studies in our past studies, we have seen that lncRNAs are highly nuclear in their expression, and hence we expected more detection of lncRNAs in a single nuclei setup as compared to single cell. And that's what actually happened. So we have about 15,000 nuclei that we investigated, resulting in about 3,000 lncRNAs that were actually perturbed across the TCDD condition. And most of them were showing that kind of difference in the central hepatocytes as opposed to any other cell type. And the reason is very straightforward, that TCDD is xenobiotic, and the central region is actually the region responsible for xenobiotic metabolism. Hence, we see that happening. And then we also again see a major shift in zonation with about 400, 500 genes roughly that show zonation, and those genes are actually functionally characterized for lipid metabolism, oxidoreductase processes, etc. So those are the pathways that are associated with zonation, and they're also being perturbed in that process. Now, with this particular data set, we also went a little bit in detail about understanding cell-to-cell communication. So we are using here CellChat, which is basically a ligand receptor analysis technique, which has their own curated base of about 2,000 validated interactions from mouse liver and from mouse per se. And then here we are basically modeling communication probability. So what you would be seeing here is a plot like this, where each dot represents the cell, and the size represents the cell size, that means how many cells you have in the cluster as opposed to the back. And any kind of interaction from one end to the other, it's a directed graph, will show where the ligand is as opposed to the receptor on that other cell type. And when we compare the control versus TCDD, we found about 843 interactions in TCDD that was previously just 200 in control. That means there were more new interactions that were happening between these different cell types when we exposed a mouse liver to TCDD condition. And some of the key giveaways here is that we see a decrease in connections for growth signaling in TCDD as compared to control, except for PDGF signaling, which basically increased in expression. And what was striking here was a major increase of extracellular based signaling pathways like collagen, VPN, laminin signaling, etc., were dramatically increased in TCDD as compared to control. And the explanation for that is that these are the signaling pathways responsible for extracellular matrix deposition that eventually leads to fibrosis. Finally, as I mentioned, there's also this piece of lncRNAs where we do not know what they do, and we know that they are essential regulators. So what we're trying to do is infer their function using a network analysis or gene regulatory network construction. So for this purpose, I'm using a big scale framework, which basically is a framework that performs iterative differential expression analysis between pairs of clusters using a Z-score, which then eventually helps us to calculate correlation values, which is apparently more stable than a regular raw count for single cell, which has a lot of, which is basically more sparse as a matrix. And then for network edges filtering, we're using adaptive threshold correlation threshold. That means you're looking at the top 5% or top 10% correlation values rather than being restricted to a correlation value. And then finally, what we're looking at here is any gene that makes connection with the lncRNA or neurotranscription factor remains part of that network. And we use gene centrality metrics such as page rank centrality, degree, betweenness, and closeness as features to understand essential nodes in that whole network. And what I mean by that is demonstrated in this example, where you have a Nash network created from that particular data set that I was talking about previously. And what you're seeing here in the center is basically lncRNAs that are not only just part of these functional modules that are enriched for specific function, but they're also essential regulators because of the gene centrality metric rank that has been calculated for them. And some of them are associated with angiogenesis and other metabolic pathways. So this helps us to accomplish two aims. Number one is to identify essential interesting incarnates from the 48,000 set that I talked about, which can then be sent out for experimental validation. But also the fact that many of those incarnates that are part of this module, and it's hard to see them here, are that they are being co-expressed for that function module. So you can infer a function based on identified association principle that, okay, this incarnate might be associated with cell division because it connects with a lot of cell division-based genes. But this, again, as I said, correlation does not mean causation. So we are trying to, and all these network connections edges can have its own difficulties because some of them might be actual targets or not. So we went a step further for those incarnates that you see in the middle as those yellow triangles. We further take them away and we see if they can form this triplex, which is basically a sequence-based approach. And the rationale for this is that they actually find that there are triplexes that these incarnates can actually form with the DNA, and these are basically hoosting bonds, which are known to show, in a way, that many of the incarnates actually regulate any downstream gene expression. And one of the examples exemplified here is of Fender, which actually forms that kind of triplex interaction with PRC to basically have a repressive mark, which is K27 triangulation. So that was our motivation for the incarnates that we found from big scale to actually further compress down the potential protein coding gene targets. And what we see here is that we found a lot of specific targets for incarnate, but then there are also shaped targets between these incarnates, which basically essentially suggest a regulatory crosstalk. So in conclusion, when we discovered a comprehensive set of mouse liver incarnates that was used as a reference for the analysis of the transcriptome of single cells, and many of these incarnates were expressed in disease conditions such as Nash, fibrosis, and PCDD-exposed livers. We saw some of these incarnates as essential markers for NAMs and also markers for myofibroblast in fibrosis. We saw an increase in cell-to-cell communication post-PCDD exposure, and we identified several regulatory incarnates that were identified in both healthy and disease condition. And we finally used RNA-DNA triplex binding interaction to find more putative targets for these incarnates that were essential or crucial for that. And the future work, we are actually working with a multi-ohm single cell plus ATAC-CK to further investigate the epigenetics behind all these interactions that we're seeing currently with single cell. And that's where we're going with that. With that, I would like to thank members of my lab, my advisor, Dr. David Paxman, some of the past members who are actually present here currently right now. Thank you for being here. If you guys have any further questions outside the session, please stop by at my poster C7 tomorrow. And with that, if you have any questions, I'd like to take them. Thank you.
Thank you very much. Any questions from the audience?
Yeah, thank you. Great talk. I just had a quick question.
Sure.
You mentioned a lot about the long-coding RNAs that shifted from the peripheral to the pericentral region. Did any long-coding RNAs show up that were like specific to the pre-locular and hepatocytes? I just asked because a lot of, it's been shown so recently that a lot of those pre-locular hepatocytes are specific in the proliferation stages for the after liver injury.
Sure, you're absolutely right about that, and I'm sorry, it was a little beyond the scope of this particular presentation, but I made a talk, I think 2 years back in the same conference here at COSI, where I actually showed liver zonation and hepatocytes and epithelial cells in healthy liver, which expanded from pericentral to midlobular to periportal. So we do have like about, I would say about easily 200 incarnates that I can think of from that study that were midlobular, and that they are present in that.
So thank you for the talk. Very impressive amount of work and data that you're producing here. So I was listening to this and all the analysis that followed, and I was thinking, how are you planning to follow all this large-scale analysis to understand the function
Sure.
and figure that out? So in some follow-up. And the other general question that I was sort of wondering in the back of my mind as I was listening to this is, this was mouse data for Nash, and what do we know about those lncRNAs and how they translate to human?
Well, both of them are great questions and something that we have thought about. So this particular study, as I said, it started with about 48,000 incarnates, and we do not know their function. So this particular piece of the study helped us to aggregate them in different contexts. So now we know for about at least about 4,000 of those incarnates that they are either cell specific in the expression, they're associated with the disease condition. So that's one kind of annotation. And then we use an inference model, and the reason for doing that is that we have a whole CRISPR group in our experimental lab that is actually actively working on characterization of their functions. And we do not have high throughput CRISPR just yet, so they are working with very specific CRISPR-off technology, and they want mutative targets. So this study also helps me to feed that back, that information back to the experimental lab to say, okay, maybe these 25 incarnates that are part of this network associated with it, quantum metabolism or cell division, etc., can be a good test for them to validate just what is now currently happening. So that's that. Apart from that, not part of the study, but follow-up of this is that for these 48,000 incarnates, I'm also doing an ortholog analysis to actually see which of them actually have human orthologs with other, mostly human orthologs, because humans highly well studied in terms of incarnates too, to basically connect and add that translational piece. But since our lab itself has animal model as mouse, we focus more on that first, even though mouse are less interested than human. But I think the ortholog analysis would help us to add that context to some of the incarnates that might show up.